Start by quickly reviewing the outline of my talk. So I'll give a bit of background on what structural variants are, what was motivating this challenge before giving an overview of the data that I'll be analyzing. And as Paul mentioned, I'll be reviewing some of our scoring approaches, talking about also ensembles with some of the crowd analysis, and closing off with a discussion of error characteristics of the different approaches, followed by conclusions. So we're interested in structural variations, which are rearrangements of medium to large size segments of the genome. They are defined by at least one double-stranded breakpoint and adjacencies of the resulting break ends. So for our challenges, we're specifically focused on the following types of structural variants. So for example, you can see, in the deletion, there are breakpoints between A and B and between B and C, and the adjacency is between the downstream break end of A and then the upstream break end of C. Now, SVs are associated with several diseases, including cancer, and for example, I'm showing here a rearrangement that's associated with chronic myelogenous leukemia that's characterized by two breakpoints, one in chromosome 9, another in chromosome 22, and the resulting fragments join together to form the BCR-ABL fusion gene. This is interesting because it represents a disease-specific druggable target, and in fact, drugs such as Gleevec, also known as imatinib, have been developed to target this drug. So in addition to revealing or providing a better understanding of the mechanisms of different diseases, identifying structural variants can help provide a real impact in the development of novel therapies. However, despite their importance, identifying them in sequence data is a hard problem. As Paul mentioned previously, different approaches to identifying them have very little overlap, so it's a good problem to have for a crowdsourced challenge. So Paul mentioned BAM surgeon previously, and it was also used to simulate tumors with structural variants. So because it's a simulation, we then know the breakpoints of the spiked in mutations. Each challenge specifies predicted structural variant breakpoints for a single tumor. So I'll briefly go over the tumors that I'm going to be focusing on. The first two tumors have on the order of 300, 600 structural variants, and the last tumor has an order of magnitude more. The cellularity, as in mixture of normal and tumor data, is 100% for two of them, but 80% for the second tumor. The types of structural variants is slightly different for the second and third tumor with insertions included. And finally, there are roughly 70 to 50 submissions per tumor. And since different teams were allowed to make multiple submissions, we can summarize that by saying there are about roughly 10 teams submitting for each of these tumors. Okay, so a key question is, which submissions are the most accurate? And to answer this question, we need to know, we need a means of measuring accuracy, so we use the F-score. But to compute the F-score, we then need to have a means of identifying true positives and false positives. So our approach is to say a call is a predicted structural variant, we call a predicted structural variant a true positive if it is sufficiently similar to a spiked in structural variant. Otherwise, it's called a false positive. But then this raises the question, how do we measure the similarity between a predicted structural variant and a spiked in structural variant? So we looked at a few different approaches. First that I'll describe basically involves the intersection or overlap of the regions, genomic regions defined by these structural variants. So, for example, if you imagine that the blue segment represents a predicted structural variant and then the pink one represents the spiked in structural variant, we can compute the Jacquard index, which is a measure of the overlap. And this approach is used on the online leaderboards. Another approach focuses on the breakpoints. So if we look at the spiked in structural variant in pink, we can allow a window around each break point where it would be acceptable for the predicted breakpoints to fall. So if the breakpoints of the predicted breakpoint fall within those windows, then we would call it a true positive. And finally, I'll talk briefly about a breakpoint length distance, which I define as follows. It's a bit of a hybrid of the above two approaches. The first component of this formula involves the distance between the predicted break ends, either the start or end, and the break end of the spiked in structural variant. And it also takes into account the difference in length between the two structural variants. And it's basically an average of these two measures. Okay, so if we keep that distance formula in mind, this heat map shows distances between spiked in mutations along the rows and predicted structural variants from different submissions. So each column represents a different submission. And the darker colors indicate a closer proximity between a spiked in mutation and a predicted mutation. And then along here, you can see the team membership associated with each submission. So for this heat map is for the first tumor with 100% cellularity, and you could say maybe about 80% of the spiked in mutations are covered by predicted structural variants, and it does vary based on submission. If we then look at the second tumor, which has instead 80% cellularity, then you can see that a smaller fraction of the spiked in mutations are covered by these predicted structural variants, and this is expected due to the difficulty of the tumor since the signal is essentially lower. Now, if we look at the breakpoint closeness type of evaluation, to reiterate, we would call a predicted breakpoint, or we would call a predicted structural variant a true positive if the break-ends fall within the windows around the spiked in variance break-ends, and we allow a certain flank amount, and this flank amount we can vary and see how the results change. So here I'm summarizing the results where the color of the background indicates the F-score, so a measure of the overall performance, and along the rows we have the different teams that participated in the first challenge with the tumor with 100% cellularity. And along the columns, we see the results from different flanking amounts. So we can see from when the flank amount is allowed to be just one base pair, the F-scores tend to be lower, and as we increase the flank amount around the break-ends, then the F-scores tend to increase. However, we also note that the rankings, they do change a bit as you go from different flanking thresholds. And when we look at the second tumor, we see some similar results where when we increase the flank amount, then the F-scores tend to increase as well. And the rankings can change somewhat when you change the flanking threshold. Okay, but we again ask the question of whether or not we can exploit the wisdom of the crowd to improve performance. In other words, can we combine different submissions produced by different pipelines such that the performance is improved? So for example, we can define an ensemble based on majority vote. If we have 3 pipelines, then we would look for calls that agree or calls that are apparent in at least 2 out of the 3 pipelines. So this SV region is covered by all 3 pipelines. here, and so it's included in the ensemble set. This one is only covered by 2, but that's still a majority, so it's also included. This region is only identified by pipeline 2, and therefore it will not be included in the ensemble set. And this indicates that we potentially reduce the number of false positives because we focus on structural variants agreed upon by multiple pipelines. And because we're looking at agreement between multiple pipelines, we have support from multiple approaches, and that could increase our number of true positives. Okay, so as Paul presented, the SNB submissions show that we can create ensembles of them that perform about as well as the best submission if we, at the, say, 50th rank, combine the top 50 submissions in an ensemble, then it performs better than the individual submission at rank 50. When we look at SVs, the picture is different. So we could see that the ensembles indicated by the triangles, they perform less well pretty early on in the ranks. So again, if we look at the third rank here, the ensemble of the top 3 submissions performs as well as the best submission, but if we move along to around just before the 10th rank, then there's already a drop-off in the performance from the ensembles. So if we try to break that down a bit into how the different ensembles are performing in terms of recall and precision, we can see that the ensembles, their recall is really defining their overall performance here. So we see, again, a drop-off pretty early on, whereas precision remains basically equivalent or slightly better than the best individual submission throughout. And so there are still benefits to including an ensemble, but sensitivity is sacrificed. So taking a majority vote to define an ensemble is perhaps it could be considered a naive approach. So if we instead considered the errors of the different methods, we might be able to, in the future, devise a better ensemble approach. That is why we also looked at error characteristics of the different submissions. So what that means is we assessed the features of sequences around structural variant breakpoints, and we tested the association between these features and false positives or false negatives. For example, for each team, we trained random forests to classify breakpoints as erroneous or otherwise. And because we use random forests, they produce variance importance measures that are values that measure the strength of these associations that we're interested in. And then the direction of the association we determine by a comparison of the sequence feature values for erroneous versus non-erroneous breakpoints. Okay, so this is a bit complicated, but I can, if we focus on the left plot first, we have along the rows the different teams participating in the challenge for the first tumor, and along the columns we have the different sequence features that we examined. The size of each dot reflects the variable importance, and then if the dots are blue, that indicates that a low value is associated with false positives in this case, and if it's red, then higher values are associated with the false positives. But what we can see here is that there are some features that are, such as mapping quality, which are associated with several errors, which are strongly associated with errors for several teams, but we also see some teams that have distinct error profiles. And similarly, when we look at the results for the third in silico tumor, we have, again, mapping quality appears to be associated, low mapping quality to be more specific, is associated with false positives for most teams, but there are some teams that produce more distinct error profiles. And then if we look at the false negative associations, we see features like strand bias, which are positively associated with false negatives in both the first and third tumors. But however, there tends to be more agreement across the different teams. Okay, so we can also summarize our error associations in a plot like this, where the columns represent the different tumors, and we have the different sequence features along the rows. And instead, we're showing the correlation between sequence feature values and the number of teams that had an error at those specific breakpoints. And we can see that there are differences from tumor to tumor, such as here with tumor coverage, but there is a bit of agreement sometimes, such as strand bias is positively associated with error in all three challenges. Okay, so to summarize, the overall performance is lower for tumors with lower cellularity, and the pipelines perform, there are pipelines that call SVs fairly accurately. If you recall the heat maps, the distances are not that far between spiked in mutation breakpoints and predicted breakpoints, but the rankings can vary based on the scoring method, as I showed, based on the flank amount threshold. And although it's possible to see performance improvements of ensembles over best submissions individually, they're really minimal, and it depends on how many you include in the ensemble. Furthermore, we saw that different teams have different error profiles, and but we also saw specific associations that are more general, such as structural variant false positives and false negatives are associated with low mapping quality and high strand bias, respectively. So I'd like to finish by thanking my supervisor, Paul, as well as key members of the lab that helped, such as Katie, who was mentioned earlier, because of her work on the SNV analysis, which carried over, and collaborators that co-organized the challenge, particularly Adam Ewing, who Paul mentioned already, who developed BAM Surge and the simulator. Okay, thank you. Introduce? Okay, I guess we don't have time for questions. So I'll introduce now Ken Chen, who is from MD Anderson, and he developed with his group NovaBreak, one of the winning algorithms for predicting structural variants.
Okay, so thank you. Thanks very much. Thanks, Paul and Hannah, for the opportunity to come here to to share with you our experience of developing a new structural variation coding algorithm and how the crowdsourcing and Dream Challenge has helped us sort of make algorithm improvement over